Good afternoon and welcome to Agri-Food Conversations brought to you by iSelect Fund, the Van Trump Report, the Yield Lab Institute, and Family Farms Group. My name is Tom Bunn, an associate on the iSelect Fund Ventures team, and I'm excited to welcome you all to our discussion today. As many of you know, Agri-Food Conversations is all about driving innovation in food and ag. Each month we highlight a specific theme, and this month's theme is soil carbon. And on today's call, we are joined by Dr. Barry Goldman, co-founder and chief scientific officer of Pluton Biosciences. Pluton Biosciences extracts valuable natural resources from the soil in the form of bacteria, fungi, and viruses, using its innovative platform to quickly and cost-effectively mine valuable microbes for use in next-generation products that solve real-world problems. The company's technology discovers novel microbes to use in new natural products in months, not years, and at a fraction of the cost expended by current labs. However, it is now turning its attention to how these microbes can help improve soil carbon. Each of you knows companies are more likely to succeed with the right network of customers, talent, investors, and advisors. We've invited you to this call because you are some of the smartest and most talented people in Pluton's market. You are potential customers for their products and services. You've built a company or worked for a company similar to theirs, or you are a sophisticated businessman, business person, or agricultural professional who understands the market and the challenges and opportunities that Pluton may face. Before we get started, we have a quick poll question to get a better idea of who we have on the call today. Please take a few seconds to answer. A few process comments while the poll is running. We are not soliciting investment in any way whatsoever. This presentation is to provide information to help Pluton find customers, mentors, or other strategic relationships that can help Barry and the Pluton business grow the business. Secondly, you were all on mute. However, you can use the chat window to ask a question at any time, and we will have a dedicated Q&A section during which you can raise your hand and I can unmute you, or we can unmute you, which is the best course of action. However, if you don't want to raise your hand, you can type into the Q&A box, not the chat box, the Q&A box, to type your question there, and we'll get to them in the order in which they came. Finally, this presentation is being recorded and will be available for replay. So without further delay, I am pleased to introduce Dr. Barry Goldman, co-founder and CSO of Pluton Biosciences. Take it away, Dr. Goldman.
All right. There's the forward-looking statements. And so thank you for inviting me. Again, I'm Dr. Barry Goldman. My background is in microbiology and computational biology. I spent 18 years at the Monsanto company, leading various teams in computational biology, tech scouting, microbiology. I was recruited to Indigo Ag in 2016 to lead and build their discovery program. I eventually led all of their research. And then about a year and a half ago, I left Indigo to really get Pluton up and running. The whole premise of Pluton is the idea is that you can't imagine it, a microbe already does it. There are 1 trillion species of microbes in the world, and we've just uncovered that knowledge. Yet the technology to find that is still back 25 years ago, but as we look at one microbe at a time. Pluton was built with the idea that we could leverage and identify these next generation microbes we don't even know what they do, but we can imagine them much faster and much more cheaply. So if we think back to the dawn of man has used microbes to make beer and to make wine and to use lots of processing. But in the beginning of the 20th century, they started turning the idea to other ways microbes could help. Fleming understanding antibiotics. Monsanto and groups like Monsanto using genes to give resistance to insects or tolerance to herbicides. And more recently, the discovery of PCR and DNA sequencing and the development of diagnostics. Now, the critical thing about this is these were done with just a handful of organisms, and it's because we didn't really know how much was out there or how to find them. But massive sequencing has dramatically changed that. So 25 years ago, the first 2 microbes were sequenced. 2. That was 25 years ago. Today, last year in 2020, we sequenced over 300,000 microbes. That expansion of data has been phenomenal. Now, all those few more organisms generated $1 trillion, over $1 trillion in revenue. Yet we're still thinking about that as one organism at a time. Again, because of sequencing and computational methods, how we find them has changed. So if you go back and think about how microbes were identified and how they're working today, companies come out and they pull one microbe out and they sequence one at a time. And maybe they sequence 10 or 50,000. But if there are trillion species of microbes, how do we get access to them? So again, that is what Pluton is trying to solve. Now we think about it this way. Traditional sequencing, which looks at one microbe at a time, is an analog function. It's like looking through the yellow pages. If you can apply massive sequencing and massive computational methods, and instead of looking at one at a time, you look at populations at a time, that is hundreds or thousands of microbes at a time, you can look much faster than that. So there are companies today, and they talk about on their web pages, we have 50,000 microbes in our collection. And if you look under the covers and you say, how many products do they have? Is they have 2 products. And I submit to you, when you have 2 products, you have 2 microbes and wasted effort on 49,998 microbes. If you're only looking for the microbes that do what you need to do, and isolating and freezing them and sequencing them and using bioinformatics, you've saved a lot of time, you've saved a lot of effort. And because you can only look at 50,000 per year, you're basically moving very, very slowly. You're using the yellow pages. But if you can look at billions per year, you can move much, much faster. So let me tell you what we do more specifically. We start with environmental populations, and in every teaspoon of soil, there's 10 to 100,000 different microbes. So companies say we have 50,000 microbes. If you go to go outside your home and you pick up some dirt, you have 50,000 microbes. It's not the microbes, it's how do you find them? And so we test them looking for populations against a targeted assay. In our proof of concept, we went to find things that killed insects. And in 6 months, with one full-time person, we walked into an empty lab, and in 6 months, we found 10 novel microbes that could kill that insect. That's a revolutionary way to get access to that. We found 2 of them had never been seen or associated with any insecticidal activity before. When people ask, what is the next, and the revolutionary microbe in agriculture is Bacillus thuringiensis, but we're now using many of those. And we're looking for a new one. If you'd ask, where's the next Bacillus thuringiensis going to come from? It will come from methods like ours. So those assays allow us to rapidly find novel discoveries, which is great, but those aren't products yet. So now to make products, we take those environmental samples and populations and we sequence them, which sequencing and the technology now enables us to do cheaply and rapidly. So we use computational biology. We now have tens of millions of knowledge in our database. And so instead of moving one product forward, which was traditionally done, now we can go to our database and say, hey, I found this really cool rare guy. Do I have more of them? And now I can look at my database. Instead of pulling out one, I can pull out hundreds. And so just like a pharma library, we can pull out microbes that are similar, but not quite the same, to put more and more of these products into the pipeline. And that does 2 things. It makes our products to market much faster because we're putting a bunch of different things into our testing pipeline that are better. The second thing it does is it expands our IP and allows us to lock up a new market by having that IP. Now we generate 3 products. We generate biologicals, that is the microbes you apply, and this is a $5 to $6 billion market and the fastest growing part of the ag market today. We identify metabolites that are small molecules, which we believe can be used to augment or replace synthetic chemistries. That today is a $50 billion industry. And finally, because we were sequencing so much, we can identify novel genes, which can be put into as plant traits to add to transgenic organisms, to add them to those pipelines. That's a $15 billion industry. Together, this is an over $70 billion industry. Here's the first products that we're trying to get to. We're working on a pesticide, which we've moved through. We're moving into formulation right now, and we hope to have onto the market in 2022. In addition, and I'll talk about this in just the next few slides, we're working on a carbon capture product. We are partnering with a big ag, and we're into the validation phase today. And finally, we're working on a fungicide, which is a unique perspective. The first two are biologicals. The third is a small molecule. And the interesting thing about this fungicide is we are working with a partner to essentially inject the active ingredient into trees that are dealing with this fungi. That's the idea. It is a new idea, but we're working with novel partners to get there. All right, so let me tell you things that we've been thinking about for a while. Agriculture feeds the 8 billion plus people in the world. It's critical for us to grow food in a sustainable and nutritious ways. One of the problems with ag is it emits greenhouse gases, as do other parts of the industry. And in fact, since 1958, we've seen an increase to 416 parts per million of CO2. That has having a dramatic impact on our culture and our civilization. In addition, growers who are working on the land and are trying to stay afloat are not seeing a lot of return on their investment. And so we think about, is there a way that we can figure out to mitigate CO2 and the huge amounts of land that are present, bring CO2 out of the atmosphere onto growers' lands and generate new revenue sources for growers. So we're of course not the first company to be thinking about that. What we believe, we're approaching it in a novel way. Okay. So what we're driving toward is an idea of carbon sequestration. Now, as it turns out, that has been in the news an amazing amount for somebody who's been thinking about this and searching forever to try and find articles on this. All I have to do is, Google search, and I'll see 1,000 things in the last 2 weeks. So the nature of this has changed dramatically in the last month or so. It's been quite interesting. But as we think about carbon sequestration, we can think about ways to reduce atmospheric carbon. In addition, because you're putting it on the land and because carbon can go into the land, you can also think about ways of increasing food production. Now, what we are doing differently is that we're thinking about how microbes can be done to sequester carbon. Now, microbes themselves can only work so-so. So a lot of people know if you walk onto a farm that's really well managed, in the springtime, you'll see a green layer on top of the soil. Those are called cyanobacteria, and they photosynthesize. They take energy from the sun, they take CO2, and they make sugar. They're great, they're amazing. It's a sign of a good farm when you see that green on top of it. The problem they do is they run out of nitrogen. Turns out they can only grow for so long, and they only live near sunlight. And we thought, well, what if we can take organisms, microbial organisms, that take sunlight not just at the surface, but maybe a little below the surface and a little below the surface and a little below the surface. That is at different depths and different wavelengths, and there are microbes that do that. Can we improve that sequestration? The second thing we thought about was, well, that's okay, but they run out of nitrogen. So we said, maybe we can put them with organisms that also fix nitrogen. And you know, fixing nitrogen from soybean and the rhizobium that are associated with the nodules in soybean that fix nitrogen and give it to the plant in exchange for carbon. We thought, what if we could do the same thing? That is, put microbes that photosynthesize with microbes that fix nitrogen and have them cross-feed each other. That is what we're trying to do and identify microbes that can do that, looking at the diversity of microbes that are out there. So it's a little bit of a complicated slide. We start with our samples, our soil samples. We do different kinds of what are called selections, ways to grow them as populations. And we ask for them to fix both nitrogen and photosynthesized carbon dioxide. And there are ways we can do that. You can come by the labs and see how we put lights on them, etc. The calculations we have done from this are that we can pull out 1.7 tons of carbon per acre. And again, what we're doing requires that you get it onto a lot of acreage. With 300 million row crops, we believe we can take out 500 million acres, 500 million tons of CO2 per year. That represents roughly 5% of the CO2 emitted per year. Currently, Indigo and companies like this are buying that at 15 tons per acre, and there's a market. Indigo has close to 1 million acres signed up for this. That represents roughly the $2 to $3 billion worth of revenue per year. We, again, have signed with a big ag partner. We own all the IP on this. Right now, they're just paying us for a right to have access and negotiation with us. We are identifying 10 consortia that sequester carbon, really improve through also fixing nitrogen. And we expect to be doing testing in the controlled conditions. So let me tell you the state of the art today. So there are 3 ways to do this. There's improvement in biomass, there's cover crops, and what we're calling micromining through consortia. The biomass is putting microbes onto seeds, and that would increase their biomass, would put roots into the soil. This is a great way to increase biomass. It's a little bit cumbersome. It's a little hard to put the microbes onto the seeds. Not every grower is set up to do that. And it tends to have a short-term impact. Cover crops. Now, cover crops do exactly what they're supposed to do, but they're a pain. They really bring in nitrogen and carbon. They store, protect the soil and runoff. They store and save water, but they're expensive. And they're very time intensive to bring to market and to deliver on those products. So as a consequence, not a lot of growers are adopting these practices. Because we have a microbial product, we can do 2 applications in pre-season and post-season. You can spray it out the back with the same things you use the chemistries from. You can get both carbon and nitrogen. It also introduces microbial diversity. We are predicting that we can bring this out for $1 to $2 an acre, and it should be no extra work for the grower. If you continue to do this year after year, there will be long-term impacts. All right, I'll summarize now and be open to and leave it open for questions. So Pluton combines new discoveries. We integrate massive sequencing with computational, and we believe we can do this 100 times faster and again, protect our freedom to operate. We're now working on a carbon sequestration that will sequester CO2 by combining it with nitrogen fixers, and by doing that, provide grower revenue. And right now, this work is being sponsored by a large ag company. We believe it'll be ready for market by late 2023. And I thank you.
Excellent. Well, Dr. Goldman, thanks so much for walking us through what you're working on at Pluton. We will now proceed to the Q&A section. So again, if you have a question, it'd be ideal if you could raise your hand and we can unmute you. Just it's a little bit more efficient. But otherwise, please go to the Q&A pane and you can type your questions in there and we can read them out loud to Dr. Goldman. But I guess, Dr. Goldman, to get started, would love to hear kind of your take on the industry as a whole. Obviously, you mentioned the last few weeks there've been a deluge of articles about this. Would be curious to get your thoughts on why now, and it seems great timing with what the infrastructure you've built with Pluton, but also would love to hear kind of the competitive set and what you think the landscape is out there with any competing technologies or even different routes to do the same thing.
Yeah, so why now? I think the election of a Democrat who is more open to to promoting this among growers and kind of coming out with a way. And he's very much said this and talked about climate as a driving force. So the political winds are working in that direction. I think that's kind of one of the biggest impacts. But the need to, and then as we see weather where you have a polar vortex, and we're seeing the worst, coldest weather in the Midwest that we've seen in 30 years. It's on top of mind. Last summer we saw fires. I was actually out in San Francisco when I got up in the morning and I couldn't see across the street. In Australia last year we were seeing heat and fires that were unparalleled. So I think people are seeing that impact and saying, is there something we can do? And with a change in administrations that is more open to that, I think they're moving in that direction. So I think that's kind of the political winds that are blowing in this direction. In addition, the first grower received a check for $115,000 for mitigating carbon. I think that was kind of, I think a lot of growers, and I've talked to quite a few growers, were saying, all right, I'm willing to try this because it'll improve my farm. But at the end of the day, I better get money from this. I mean, I've really got to get money. This can't just be me making my farm better, because there's other ways I can make my farm better. And putting a bunch of things in the ground, if that's more work, I'm not really excited about it. And so, watching those first growers start getting money from the space, in addition, more companies are buying into what Indigo is doing. You just saw Bayer recently saying that they're going to figure out ways to do this. There are startups who are trying to figure out, how can we measure the soil without actually going in and doing a test on this? How do I not have to pull soil out of the ground and then find the absolute amount of carbon and nitrogen that are there? So there's a lot of things that are happening in this space that are sort of converging at one time.
Excellent, thank you. Looks like Kyle Mayman has a question. Kyle, looks like you are unmuted.
Yeah, I just had a quick question. So maybe I didn't quite understand, but with this microbial technology, what would be the, I guess, what would be the X plus, or how much more sequestration would we expect than just the common practices of reduced tillage or no-till systems plus cover crops? How many more tons could we expect, or as a percentage? I'm just trying to understand that. Or would you do this in lieu of those practices?
I think you do that in addition. I mean, that we're expecting it's in addition. We have calculated 1.7 tons of CO2 per acre increase. I can go through all the calculations. That turns into roughly a half ton per acre of carbon. That's the difference of molecular weight of CO2 versus carbon itself.
So in some cases, it could double it then, really.
We think it can be quite beneficial, and it's because microbes can grow very fast on very little nutrients. And because they're cross-feeding each other with carbon and nitrogen, again, because we're starting with consortia, a group of microbes, we believe that it could, that they could grow very rapidly and put more carbon back into the soil.
Great, thank you both.
Thank you.
Prim Warrior asks, understood you are working with a large ag company. Are there opportunities for additional collaboration / partners?
Yes, there are. We're actually working on another carbon product that I'm not at the moment at liberty to divulge. It's a little early for where we are, but we are working on a second carbon sequestration product that is not the same. And so we could be discussing that as well. That's a little earlier in the stage. I didn't even put it onto the slide, but we're working with other. And some of the things we'd like to do, we've been starting to talk to different startups around how do you capture carbon and release of carbon from satellites. And is there a way to do that more quickly, more rapidly? And so that once we start getting to the field, how do we prove, I come from Monsanto and Indigo, and you have to prove what you're trying to do. So how do you actually? So I have a great idea. I believe I can show that in the lab, but at the end of the day, I got to get growers money. I have to show that our value is worth it. And we're not a satellite company, so we're looking for partners today that can help us figure out how does this really work in the way we expect. It will be very expensive to do this as carbon. Not everybody believes that, but if we can work with a partner that can show, at least to our satisfaction, that what we're doing works, that would be immensely valuable, and we can see which ones are working better, so that we can get to our first generation product will probably work. Our second generation product will work great, and that's really what we're trying to get to.
Great. Justin Perriman asks, are you partnering with any St. Louis-based ag tech companies?
Am I working with St. Louis-based ag tech companies? Well, we're working with a lot of folks in St. Louis. This is a great community to be working with. I'll leave it at that.
Luke Esch asks, what is the potential for carbon sequestration on rangeland rather than row crops?
Yeah, that's a great question. We thought about it. I think it could be really powerful. The difference is we haven't figured out a really clear way to deliver it. One of the reasons you have row crops is everybody has the tractors, they're already going out in the field. We think it's really important to not do more passes of the tractors over the field. That's why we're doing it at harvest and at planting or at burn down. Is if you send the tractor over the field yet one more time, you're mitigating a lot of what you're trying to solve. We believe rangeland could be really phenomenal, but we haven't come up with a great way yet to figure out how to deliver it such that you can get it out to where we want to, which is, again, not a lot, but all over the place. And it could be more expensive, but you know, I have to say the rangeland looks really enticing because we really believe we could increase yields of grass and improve and increase diversity on those soils. But and if you have a way, please let us know to get it out there. We've had people talk about drones and delivery via drones, but when you do the math on that, it goes, well, that might work, but I just haven't figured out if that can. If there's a way to do it where you're really not spending too much money or too much carbon dioxide to deliver it, I think it could be really powerful, but to date, we haven't quite figured that out.
Great. Dilip Shah asks, does your injectable fungicide work only on specific fungal pathogens of trees, or does it have efficacy against pathogens of high volume commodity crops?
We don't know yet. Right now, we're really targeting one specific pathogen. And Dilip, since you're an expert, you're a world expert in this space, we're really looking at an oomycete. And we think that it could have broad efficacy if we can figure it out, or at least the search to find these and the ones we're looking at could have efficacy against other oomycetes, Pythium, Phytophthora, et cetera. And so there may be other things that we can find activity against, but right now that we're in kind of an early stage and we're trying to focus.
Abby Singh asks, what are the key developments needed to make this technology successful? Yeah,
being able to grow it up cheaply. So, microbes. So the biggest issue right now in biologicals is, you've got a great microbe, you've seen it in the lab, it works. Now trying to scale it, where you're going from essentially a few liters to 5,000 to 10,000 to 50,000 liters, that is an art. There are not a lot of places where you can do that. And so making sure you have a robust culture that can grow to that scale so you can keep your costs down is a critical step in this. And so, that to me is the proof in the pudding. We can get to a certain place, but if you can't scale, then you're going to hit it. After you've scaled, then you have to worry about, is every grower doing it the right way? How do you make sure that that's applied in the right ways? Adoption is always an issue. I mean, there's a lot of ways. Discovery is very early in the process, and while we believe it has tremendous potential, that's where we are. It's potential, and we have to think about how do we get there? How do we get there fast? How do we get there right? And how do we go through all the pieces that we need to deliver it as a great product?
Great. Well, Dr. Goldman, last question I would ask you is how can this audience here, both listening now and retroactively to the recording, how can they help you, and how can they find you or your teammates at Pluton?
Yeah, so if you just look up Pluton Bio, we have a website. I'm available, and sorry I didn't put my email up there. I'm at bgoldman@plutonbio.com. You can certainly email me. We love talking to growers. I mean, you know, we do talk to some, but since we've started moving down this way, we'd love to have a few people come by the labs and start showing them what we're working on, and ask questions, and say, and just set up meetings and just say, okay, why not would you adopt this sort of product? What would prevent you? Can we use you as a way to start testing? Just questions like that.
Terrific. Well, Dr. Goldman, thanks so much for joining us today, and congratulations on all your and Pluton's progress to date.
Thank you, Tom.
I'd also like to thank the audience for their active participation. And again, we host these calls every Thursday at 3 p.m. Central. We'll have a few more in the soil carbon realm. And you can register for these upcoming agri-food conversations by going to agrifoodconversations.com. And once again, also, if you know someone that would like to watch this webinar, feel free to share it with them. A replay will be available and actually emailed to you within the next 24 hours, or they could go to agrifoodconversations.com. So thanks again, Dr. Goldman, and all in attendance, and hopefully we will see you next week.